All right, we're back, and we are going to go into detail now on these mechanisms of evolution. All right, so the first one is mutation, which we've already talked about a little bit when we talked about DNA. So a mutation is just a change in the base pairs of the DNA, right? It could be a simple substitution, just going from a T to a G. It could be a whole section inserted or a whole section deleted. So there are different types of mutations. A mutation in a non-reproductive cell, a somatic cell, will only change that one individual. It's not affecting the population, so that doesn't lead to evolution, right? So if you are exposed to extra sun and you get a mole on your skin, yeah, there may be some mutations in your DNA there, but that you did nothing evolved, right? It's not affecting your children. It doesn't get passed down that way. That would be a Lamarckian idea, that something that affected you during your lifetime then was passed down to your children. So only mutations that take place in the gametes can lead to evolution, because those are the ones that end up in the next generation and that they could then pass down as well, because it's in all of their genes, all of their cells. So how do mutations occur? First of all, mutations are random. Um, we are going to be discussing this in lab as well, but some environmental factors may increase mutation, but we can't predict how many mutations there will be. We can't predict where they will be, and we can't predict what they will be. Mutations are truly random, um, and they're rare. In gametes, they're rare, right? If we think of how many cells you have, um, a mutation in an egg or a sperm that that makes some sort of difference to it is very, very rare. But they're common enough over billions of years that they have led to all of the diversity that we see on Earth today. So they're like the mother of all genetic variation is mutation. And so when it happens, it's a very, very powerful force. Okay, the next mechanism of evolution that we're going to discuss is genetic drift. And there are two subtypes that we'll also talk about. So genetic drift is just the completely random change in allele frequencies over time due to chance. So this is something that is just, you know, rolling the dice and whatever alleles come up. If you remember when we talked about like probability and chance with Punnett squares, right? If we were doing a Punnett square with this, right? So we've got two heterozygotes here. You have a 75% chance that you are going to have some sort of dominant phenotype, right? And only 1/4 chance that the offspring will be a homozygous recessive, right? But chance, right? If you took that to a casino, that doesn't mean that you're going to win 75% of the time, right? If you're betting on the dominant phenotype, right? It's possible that if these individuals had 4 kids, all 4 could end up with this genotype, right? There's a lot of chance involved. And so genetic drift is really looking at chance. If you have a small population, chance plays a much bigger role. So we're just going to do a little exercise for a minute here. And I would like everybody, if you can take out your phone, which I know you never have near you while you're watching lectures, but take out your phone and just ask your phone to like flip a coin, okay? So I want you to flip a coin, um, flip it 2 times, okay? So this would be if you are an individual and you have a potential to give, these are your 2 alleles. You have the head allele or the tail allele, right? And when you make eggs or sperm, right, it's a coin toss what's going into that. So if you flip a coin 2 times, we are now, let's say, taking 2 individuals and determining what is the genotype of that offspring, okay? So everybody do that. Wait, pause your phone, and we'll wait a minute. I can't pause the video, so um, my guess is now, if everybody, I wish we could all write up on the board right now what you have, but my guess is that some people got um, two heads, some got two tails, and some people got heads and tails, right? So it's kind of a mix, but the chances of, right, even though it's supposed to be a 50% chance of heads and a 50% chance of tails, everybody didn't get that, right? So in a very small population, we just had two individuals here giving an allele, right? Um, the chances of getting all heads is pretty high, or chances of getting all tails is pretty high. But, and so if you have a small population of like 30 individuals breeding, an endangered species here, right? The chances of everybody getting all the same alleles in one generation, right? Or maybe one individual getting that tail allele, and then the tail allele could kind of die out. Not because it's bad, not because there's selection against it. That tail allele is completely equivalent, right? It could be whether your fur swirls to the right or swirls to the left, right? It has no effect on fitness, but it could be lost just by chance. On the other hand, if you have a population of 1 million individuals, your chance of losing that allele are much smaller just because, right, chances are it's going to be passed down. If you've got 1 million individuals, half of them have that allele to start, they'll, you know, a good portion of them, 25%-ish, are going to be passing it down. Um, so this is genetic drift, like a big picture. And now we're going to talk about those subtypes of genetic drift. So as I said, those two subtypes are the founder effect and the bottleneck effect. So, but before we get into those details, I just want to stress again that drift is not natural selection, okay? It's not anything advantageous that's either getting fixed, which means 100% of the individuals get that trait, or lost. It's just completely random. Okay, so here's an example of drift. We've got these rabbits, and in the initial population, the first generation on the left here, um, the frequency of alleles is 50% big A, 50% little a. So some individuals are homozygous dominant, only 2 of them, right? 6 of them are heterozygotes, and 2 of them are homozygous recessive. Okay, but half are big A, half are little a. Then in the next generation, just by chance, 5 of the rabbits are reproducing. Those are the ones that are circled in red. And just by chance, as they're passing on their alleles, right? It's random whether big A or little a gets passed down. We end up with 70% big A in the next generation and only 30% little a, right? So if we look at these new rabbits that are born, we have 1 homozygous recessive and we have 4 heterozygotes. Okay, so our proportion of little a has just dropped. By chance, the next year, only 2 of the rabbits in this generation have the opportunity to reproduce. Maybe it's a bad year for food, or it's really cold, and they either die or they don't have the energy to reproduce. They don't have the strength to have a pregnancy, right? Whatever it is, only 2 rabbits reproduce, and by chance, those 2 rabbits happen to be only big A alleles. It's not because the little a is bad. It's not because a white rabbit is more likely to get eaten. In this case, it is pure chance. So a common misconception of students is that recessive alleles will die out. And that's not true. Unless they're actively being selected against, recessives aren't bad. You're not going to lose the recessives. In this instance, they were lost, but it could have just as easily gone the other way. There could be another population of rabbits somewhere that lost all their big A alleles, right? If this very first generation on the left, we could have bred these 5 and ended up with 70% little a and only 30% big A, right? So I just really want to stress that this is not selection going on. Okay, so let's discuss the founder effect. This is a subtype of genetic drift. This is when a small group of individuals leaves a big population and starts their own little population. They're founding it. Okay, this happens in some humans populations, certainly when explorers would leave Europe and come and start a new colony right in North America. Those people were founding a new population, and whatever genes they took with them are the genes that are going to be in the new population. So they are not representative of everybody in Europe, right? Whatever genes they happen to have when they came over are the genes that they get. And so there might have been something at a very high frequency that was rare, or something in a low frequency that had been common. So this is common for isolated groups of people. So people who came over and didn't then mix with the people who were already here, right? But remained their own little isolated groups. It's common for species that move to islands, not like move to for vacation, but right, end up on islands. They get um, lost in a storm, and kind of birds get pushed over to an island and end up staying there, right? Or they're on a piece of driftwood that makes it to that island, or they cross, they climb up a mountain and down the other side, a small group of them, and they're now on their own. So this can eventually lead to speciation because this group is isolated, but we're not talking about that big picture yet. But just that whatever genes they've got is what they're carrying with them. An example for humans is polydactyly, and we talked about this. We had an example in lab with like um, the when we were talking about big F and little f, and the big F, the six finger gene, is actually dominant. It's just really rare. Um, but in the Amish community, it's more common. So whoever first came over from Europe in that group, they tended to have more people with that gene. So let's say it's 2% of the regular population, but I'm exaggerating here, but let's say only 10 people came over and two of them had polydactyly, right? Suddenly it's 20%. So that small group didn't represent the entire population, but now that gene is overexpressed in that population. Okay, so this is an example of the founder effect. Okay, the other subtype of genetic drift is the bottleneck effect. And this takes place when a large population is quickly reduced to a small one. So it's not that a few individuals choose to leave, it's that everybody else dies. So it could be famine or disease or some big environmental change like a flood that causes the death of a lot of individuals. Those ones that survive are not surviving because they're fitter. This is randomly by chance they survived, and they probably don't have the exact same allele frequencies as the population as a whole did. So we could think about if there is a group of deer, like a whole herd of deer, standing in a little stream on a nice day, just drinking water. Some are in the middle of the stream and some are on the banks. A flash flood comes through and wipes out anybody in the middle of the stream. So the few that had been on the banks survived. It's not because they were smarter than the others or that they had longer legs, right? When there's a flood, everybody gets flooded. They just happened not to be there. So whatever genes they have are the only genes that go on to the next generation. So here we've got a picture of a true bottle with a bottleneck. So in the original population, you can see we've got green marbles and red marbles and kind of peachy orange marbles. But there's this bottleneck event, and the ones that come through in the surviving population, we've lost red, and it looks like we have a smaller frequency of green than we did in the original. So we've changed our allele frequencies, which is evolution. So there's a few famous examples of bottleneck effects. You may have heard that cheetahs have very low genetic diversity. They're all very close related to one another. Genetic tests have indicated that you could kind of date back, like when they probably last had genetic diversity. And there's evidence that about 10,000 years ago, most cheetahs died, and we don't know why. There was some sort of event, and the current population of cheetahs dates back to that, and that probably only about 12 survived. So those 12 and whatever genes they had are all of the genes that are present in today's cheetahs. Um, and then the Galapagos penguins are another interesting example. They actually have regular bottleneck events. So during El Nino years, which happen about every 7 years, their population numbers plummet. Because the trade winds change, the ocean currents change, and their food supply changes and declines during El Nino years. So about 77% of them, up to that many, can die. Some years are worse. Some years are much better. So it's variable. But through their history, over hundreds of thousands of years, they've gone through many, many bottlenecks. So this has a large effect on their genetic diversity as well. So they've just lost some alleles that they almost certainly had a long time ago. Okay, our third type of evolution, the mechanism for evolution, is migration. And we talked about this with the tigers, right? You can bring more white tigers in, and boom, suddenly your allele frequencies in your population are different. So migration is a great way to get new genes in. This is gene flow. So it's just this movement of individuals in or out of a population. And you saw when we were doing that elephant PCR lab that individuals that, like forest and savanna elephants, looked really different genetically. That's because they didn't have this migration between them. You could easily tell who was who. As soon as you start mixing genes, you'll get alleles from one into the other. So when there's migration, usually both populations gain diversity because it's usually a two-way road. It's possible that if all individuals are migrating out of one place and into another, that one population is gaining diversity and the other's losing. And the amount of gene flow obviously depends on the organism itself and the geography of the area. So plants have gene flow is going to be usually with organisms that live a little closer to them because they can't pick up and move. Um, Species that live on islands have less gene flow with other organisms unless it's like nearby islands and it's a bird that can fly back and forth, or if they're like at the top of a mountain, right? To get down and up to the next mountain would be hard, or areas that are kind of, there's a river and it's hard to get to the other side, right? Then you may not have that gene flow. So here's an example. We've got birds here, same species, but one has only the big H allele on the left side, which makes them blue, and the little h allele is on the right side, which makes them red. And there is just a little bit of gene flow. One blue one goes over to the right and one red one goes over to the left. And our offspring, we've now introduced a new gene into each of these populations. So these populations have now evolved and their offspring can have that as well. Okay, and now we are up to natural selection, which is probably um, the mode of evolution that people are most familiar with. So I'll just do a little intro to it, and then we're going to go deeper in a little bit. But for this lecture, we'll go kind of on the surface. Okay, so before we get deep into evolution, into natural selection, you need to know there are 4 conditions for it to occur, and these are Darwin's 4 postulates. He actually figured this out, Darwin's. Okay, in order for evolution to occur, for natural selection to occur, there has to be variation within the population, right? That's true for all evolution, right? If everybody's the same, you can't select, right? You can't pick white tigers to breed instead of orange if all the tigers are the exact same color, right? You have to have variation. That variation needs to be heritable. If orange just happened because they sat in the sun a little longer, then breeding only white tigers isn't going to get you more white tigers, right? You just have to keep your tigers out of the sun. So it has to be a heritable trait. The third condition is that more offspring are born than can survive to reproduce, right? So not everybody reproduces. We saw that with those rabbits, right? 10 rabbits, but only 5 reproduced one time and only 2 reproduced the next. Everybody doesn't reproduce equally. Some have more offspring, some have less offspring. And then the fourth postulate here is that survival and reproduction is not random. The ones who are surviving and reproducing more are those that had a trait that enabled them to do that, right? So one of those variations that was inherited makes those individuals more likely to reproduce. Those are Darwin's four postulates, and you cannot have natural selection without that. So here's just a really easy example of natural selection. We've got these birds that eat beetles. Some beetles are orange, some beetles are green. It's some genetically based thing, right? Maybe the green beetles have like this genotype, right? And the orange beetles are the homozygous recessive genotype. Most of our beetles are green. The birds love the green beetles. Maybe they taste better. Maybe they stand out more in the background, right? If they live in an orange dirt area. But for whatever reason, the birds eat all the green beetles, which obviously means that even if one or two green beetles survive, mostly orange beetles are reproducing. The orange beetles are passing on their orange genes, and over time, we're going to end up with only orange beetles or mostly orange beetles. You can now imagine things might flip if orange beetles are the only food source. The birds may decide, well, I'll eat orange beetles, and maybe the orange beetles will decrease in frequency and the rare green one will increase. And a famous example that some of you may have seen in school before is with these peppered moths in England. So there's a moth here, if you can't see it. Here's our moth. They blend in very nicely on the bark of trees. During the Industrial Revolution, when there was a lot of coal burning in the cities, the trees often turned black with soot. So there's this rare mutant form that shows up every now and then. So it must be a mutation that happens kind of frequently. And sometimes there'll be a black moth. When trees are light colored, those black moths get picked off very quickly, and so they tend to stay at a very, very low frequency in the population. So this dark form was less than 1% of the individuals, right? They show up, very rarely live to reproduce, and then they get eaten. During the Industrial Revolution, these black ones did really well, and the pepper colored ones are the ones that stood out, and they got picked off. So those frequencies shifted, they completely flipped. In urban areas that had a lot of coal burning, their numbers went up to about 98%, those dark ones. So that's evolution, a change in the allele frequencies. Once air got cleaned up and trees were no longer sooty looking, those numbers shifted again, and that dark variety is once again rare. So just as I mentioned earlier when we were talking about those rabbits, recessive traits don't decrease only because they're recessive. They will decrease if they're selected against or due to chance, right, in drift, but they don't decrease just because they're recessive. So if you have a population of that's 50% big A, 50% little a, no forces of evolution going on, no drift, no selection, that recessive trait will stay, and it will stay at 50%. So if there's no evolution going on, there's no selection, there's no drift, there's no migration, there's no mutation, the allele frequencies will stay the same, which is basically saying they're not evolving, right? If evolution is a change in allele frequencies, then we could say if there's no evolution going on, there won't be a change in allele frequencies. So recessive won't disappear just because it's recessive. It's going to stay there in the same frequencies. And we will stop here and pick up more natural selection in a little bit.